Okay, let us now move on to our next question. So this question is very similar to the previous question that we did. However, instead of looking at just an amino acid, we are now looking at a peptide. So the first thing that we need to do is obviously read the question carefully. So what the question is stating is to calculate the charge and the isoelectric point of the following peptide at a pH of 3, 7, and 12. Okay, so we have our peptide here. So the first thing that we want to do is translate it from a 1-letter code to a 3-letter code. Now, is this strictly necessary? Not really. But what I find this is good for is to make sure we don't make any silly mistakes in terms of accidentally looking at the 1-letter code and sort of thinking it's a different amino acid. So D is aspartate, S is serine, Y is tyrosine, L is leucine, K is lysine, and C is cysteine. So now that we've got our names written in, the next thing I do is to draw a table. Now, immediately upon drawing my table, what I will do is I include the name of all of my amino acids, but then I immediately ensure that I put in the quaternary ammonium group on the left and the carboxylic acid on the right. Why? These are our terminal groups. So this quaternary ammonium group that I've written over here, that is attached to this aspartate. This carboxylic acid, that is the terminal carboxylic acid group for this cysteine molecule. Now, again, the big reason why I do this is to ensure I don't forget them. Now, writing all these amino acids in a table just helps to keep us organized and helps to make it easier for us to work through this question. So then the next thing that we need to do once we've drawn our table is to have a look at the amino acid properties table and to have a look at what the pKa values are for these amino acids. Because if we are looking at these amino acids in a protein, there are only 3 areas in which we can see a change in charge. The first one is our terminal quaternary ammonium group. The second is our terminal carboxylic acid group. And the third one are ionizable R groups, because remember, when a peptide bond is formed, it no longer has the ability to lose or gain hydrogens. So what I have done here, by the power of editing, what I have done here is taken this table, but underneath it, I have just written down the pKa value for that group. So for example, if we are looking at tyrosine down here, so tyrosine, it has a pKa of 10.07. So what I'm referring to there is that tyrosine's R group, okay, the side chain has a pKa of 10.07. So the next thing that we need to ask ourselves by looking at this table is, do all of these amino acids have ionizable R groups? So looking at serine and leucine, those R groups are non-ionizable. So what this means is we can cross them out. We don't need to consider them in our calculations of the charge or the isoelectric point purely because they are unable to change either factors. So the next thing that we need to do is we need to compare our pKa to the pH, because remember, when the pH equals the pKa, 50% of the molecule is protonated, 50% of the molecule is deprotonated. Starting with our terminal quaternary ammonium group, we can see pH 3 when compared to a pH of 9.6. So this is a much more acidic environment, which means that this quaternary ammonium group is going to stay protonated. Because remember, we also have that +-1 buffering zone. Quaternary ammonium group is positive, so this is going to remain as +1. pH 7, again, is much lower than a pH of 9.6, so this is also going to be +1. But when we get to pH of 12, now we see that that is significantly higher in terms of pH than the pKa. So that means that this quaternary ammonium group is going to lose a hydrogen, meaning it's going to move from NH3+, so +1, going to NH2, neutral. So the charge here is going to be 0. So keep in mind when you are doing these questions, the charge can only move by one value. Okay, it'll either be +1 or 0, or it'll be 0 or -1, depending on the group we're looking at. The next one here is aspartate. So comparing 3 and 3.65, what we are seeing here that a pH of 3 is lower, so this is going to remain protonated and it's going to be a 0. Now, a word of caution here. What actually happens is a partial dissociation, because this is not just looking at one molecule and it's sort of an all or nothing principle. What we need to remember is that there are heaps and heaps and heaps of these if we just sort of had a sample of aspartate. And it means that as we gradually move into this ±1 buffer zone, so for example, with aspartate, the pKa is 3.65. So it means that buffer zone is going to start at 2.65. It'll be 50-50, so 50% dissociated at 3.65. And then at 4.65, all of those aspartate molecules have been completely deprotonated. So this would technically have a fractional charge. However, given that we're just introducing the topic at the moment, we're just dipping our feet into the water, as it were. It is okay for us to sort of work in absolutes. So how we're going to be sort of moving through this is, if the pH is lower, I want you to assume it's protonated. If it is equal to the pKa, I want you to assume it's 50-50. And if the pH is higher than the pKa, I want you to assume it's been deprotonated. So now we compare aspartate to a pH of 7, and what we see, 3.65 compared to 7. Obviously 7 is a much higher pH, it's a lot more alkaline. So this has lost its hydrogen, which means it is now -1. Again, pH 12 versus pH 3.65, this is -1. Now, serine we don't have to worry about, so we're going to move on to tyrosine. And we're starting to see the pattern here. pH 10 versus 3, okay, the pKa here is much higher, so it's going to hold on to its hydrogen and stay neutral. pH 7 versus pH 10, pKa is higher, so it's going to be neutral. And then pKa of 10 versus 12, okay, it's finally been overthrown, and it will have a charge of -1. 10.53 versus 7 is it's not going to lose any of those hydrogens, so it's going to stay as +1. 7 versus 10, it's +1. 10 versus 12, okay, the pH is now higher, so it is going to be neutral. So as we can see, I accidentally forgot a column. So our last step here is going to be to calculate the final charge. So how do we do this? Quite simple. We just simply add them up. So we have +1, +1, -1. So that is going to be an overall charge of +1. Then we have +1, -1, which is 0. +1, which is 1, -1, which is 0. Now, straight away, I've got sort of my alarm bells are going off, because the question is asking us to also calculate the isoelectric point. So this is going to be useful for us because we know at what pH our protein is neutral in charge, or a zwitterion. So then we look at the bottom one. Let's just do it for completion's sake. -1, -1, -1, -1. That's going to be -4 charge overall. Excellent. Now we can have a look and calculate the isoelectric point. So remembering that the isoelectric point is telling us at what pH our protein here, our molecule, is neutral in charge. So what I want to do here is I am looking at a charge of 0. So I know that this isoelectric point here is going to be somewhere around 7. Now, another piece of information that we do know is what is influencing this change in charge. And we know at what points this charge is changing, because that's what our pKa tells us. So in order to do this, I'm going to look at all of my pKa values. And I am going to look at two values. I am going to pick the value that is closest to, but lower than 7, and closest to, but greater than 7. So what do I mean by that? Well, at 7, which of these pKa values is closest to but less than 7? So we have ones that are less than 7. We have 1.96 and we have 3.65. So the value that is closest to but less than 7 is going to be 3.65. Now we do the exact same thing, but we go the other way. Which value is closest to 7 but higher or greater than 7? So looking through these, we have a 9.60, okay, 10, 10.07, 10.53, so we can eliminate those because that 9 is closest, and 8.18. Aha, there is our winner. So 8.18 is closest to, but greater than 7. So we take these two values, add them up, and divide them by 2, and we get an isoelectric point of 5.9. So then we have a look and we double check this. We go, well, okay, does a isoelectric point of 5.9 make sense? And yes, it does, because we saw that at a pH of 7, this protein is neutral. And what we also saw was at 8.18 down here, that is when we saw our cysteine molecule become more negative, and that would have tipped our charge of our protein from 0 to -1. So we can verify that yes, the isoelectric point for this protein is 5.9. And our last question for the day is, what is the net charge at a neutral pH? So at a neutral pH, we can assume a pH of 7 of a tripeptide containing only alanine. So what this means is that we have 3 alanines that are joined together via a peptide bond. Okay. How does the total number of negative and positive charges change following hydrolysis of the tripeptide? Okay, so the first thing that we need to do is let's draw out alanine as a tripeptide so we can wrap our head around it. Okay, so what I have done here is I've drawn alanine 3 times, but I've drawn them down together via a peptide bond. So for reference, our peptide bond is here. So the first thing we need to do is, again, let's double check the question. So what is the net charge at neutral pH of a tripeptide containing only alanine? So what we see here, we have a -1 charge, we have a +1 charge, so the charge will be neutral. It'll be 0. Okay. Now, how does the total number of negative and positive charges change following hydrolysis? Well, at this stage, we have 1 positive charge and 1 negative charge. Okay, but when we undergo hydrolysis, that is using water to break this group apart, then what we see is 3 alanine molecules separated. Okay, so now that I've drawn my alanine molecule out 3 times, we have a look at how this question will change. So how does the total number of negative and positive charges? So if we have +1 and -1, what we see here is we have, these are all +1. So 1, 2, 3. And our -1 is also 3. So how has the total number of negative and positive charges changed following hydrolysis? Well, they increased threefold. They went from 1 to 3 because the thing is that this quaternary ammonium group and this carboxylic acid, so for instance, these two, couldn't have a charge when they were partaking in a peptide bond. So that is how, by hydrolyzing these amino acids, we see more groups that are able to lose or gain a charge. So guys, I hope that going through these questions was helpful in your studies. If you do have any questions, please don't hesitate to ask. Other than that, study hard, do your best, and I'll see you in the next one.